Welcome back to the Explainer, everyone. Get ready for a seriously high-yield session today. We're going to take a deep dive into the foundational world of bacterial classification. Specifically, we're looking at that mighty divide between gram-positive and gram-negative bacteria. And more importantly, we'll trace how those tiny, microscopic differences actually lead to absolutely terrifying clinical consequences on the wards, like septic shock. So I want you to just think about this for a second. How in the world does a simple dye, invented way back in 1870, predict total cardiovascular collapse? I know, it sounds kind of wild, right? But it's entirely true. In this explainer, we're going on a direct lab-to-clinic journey. We'll see exactly how a basic staining technique reveals the hidden structural secrets of bacteria and how those very structures literally dictate the circulatory fate of our patients in the ICU. Okay, let's dive right into this. Here's our roadmap so you can mentally organize all these high-yield facts as we go. First up, the gram stain procedure. Second, cell wall structural differences. Third, endotoxins and exotoxins. Fourth, the pathogenesis of septic shock. And finally, capsular bacteria and splenectomy. All right, section 1, the gram stain procedure. The blue versus pink binary. We really owe this entire foundational system of microbiology to just one guy, Sir Christian Graham. He's the one who established this fundamental binary way back in 1870.
Let's rapidly break down the 4 mechanical steps here. Step 1, you apply crystal violet stain on your smear and then wash it. Step 2, apply the iodine gram stain. Now at this point, all the bacteria are blue. But step 3, this is the absolutely crucial differentiator. You apply HCl and alcohol and wash it immediately. This keeps your gram-positives blue, but it completely strips the color right out of the gram-negatives, making them totally colorless. And finally, step 4, apply the carbofuscin counterstain, which you probably know is safranin. So when you actually look through that microscope lens, the 187 rule is just absolute. Gram-positive holds onto that blue, and gram-negative turns pink. You've got to burn that visual binary into your brain right now. Blue is positive, pink is negative.
Moving on to section 2, cell wall structural differences, the why behind the stain. So we know what the colors are, right? But now let's transition from the lab bench down to the microscopic level to uncover the real mystery here. Exactly why do these bacteria absorb those stains so differently? What is actually physically happening on their surfaces?
Well, tracking from the inside out, both gram-positive and gram-negative bacteria share a foundational inner lipid bilayer, the cell membrane. But moving outward to the cell wall, check out how gram-positives have this massive, thick layer of peptidoglycan. That's essentially the sponge holding onto that blue dye so tightly during the wash. Gram-negatives, on the other hand, well, they have a really thin peptidoglycan layer. But here's the absolute critical difference. The gram-negative bug makes up for it with a sneaky extra layer, an outer membrane.
Let's zoom in on that outer membrane for a second, specifically its outer layer, which is made of lipopolysaccharides. Now, this contains a lot of different lipids, but there is one massive, high-yield pearl you absolutely must know, and that's lipid A. Lipid A is the specific lipid component housed right there in the outer layer of gram-negative bacteria, and it functions directly as an endotoxin. This literally links their physical anatomy straight to their most dangerous weapon. Which brings us to section 3, endotoxins and exotoxins, the bacterial weaponry. Let's clarify the specific types of chemical warfare these bacteria are actually using against human hosts. Take a look at exactly where these toxins originate within the bacterial architecture. You can clearly see the exotoxin being deployed from deep inside the cell cytoplasm over on the left, right? While the endotoxin, it's literally built right into that extra outer wall layer on the right side. So the crucial takeaway here is this. Exotoxins are toxins released from the cytoplasm of both gram-positive and gram-negative bacteria. Think of them kind of like tiny grenades that are being manufactured inside the cell and then lobbed outward. Endotoxins, however, are strictly a gram-negative phenomenon. That lipid A we talked about, it's stuck firmly in the outer membrane of gram-negative bacteria only. It isn't just a weapon, it is part of their literal anatomy. All right, section 4, pathogenesis of septic shock, the domino effect. Now we're shifting this explainer from molecular biology over to the truly terrifying clinical presentation of a gram-negative bloodstream infection. This is where things get really critical for your patients. Let me walk you step by step through the initial trigger of this catastrophic domino effect. Step 1, a gram-negative bacteria enters the blood circulation. Step 2, that endotoxin, The lipid A we were just discussing binds with CD14 receptors on the patient's white blood cells, specifically the monocytes and lymphocytes. Step 3, this binding activates those white blood cells to release just a massive, overwhelming wave of cytokines. And once that cascade starts, that massive wave of cytokines splits into two distinct, incredibly deadly clinical pathways. Those activated white blood cells are releasing two main culprits here. Down one path, TNF-alpha causes widespread vasodilation, which leads to massive peripheral pooling of blood. On the parallel path, IL-1 ramps up the expression of receptors for platelets on endothelial cells, and that causes a massive widespread consumption of platelets and clotting factors. So why does this matter for your patient? Well, because this cellular cascade directly destroys effective circulatory volume. The peripheral pooling from TNF-alpha tanks the effective circulatory volume in the aorta, drastically decreasing blood supply and perfusion to vital organs, leading straight into septic shock. Meanwhile, the platelet consumption that's being driven by IL-1, that results in consumptive coagulopathy, or DIC. It's a total systemic collapse, and it all started from that tiny little lipid A. Almost done here. Section 5, Capsular Bacteria and Spleenectomy, a Clinical Pearl. This is absolutely critical, a ward-ready takeaway regarding patient vulnerability to specific bacteria that you are for sure going to see on your clinical rotations. You see, beyond just the cell wall, some bugs actually wear an extra suit of armor, a capsule. The key encapsulated threats that you just have to memorize are pneumococcus, Neisseria meningococcus, Haemophilus influenza, and Klebsiella. Now, notice the odd one out here. Pneumococcus is the sole gram-positive offender on this list. The rest, meningococcus, H-flu, and Klebsiella, they are all gram-negative. So, how does the human body actually handle these heavily armored capsular bugs? The spleen. The spleen provides vital opsonization, which basically means it flags, removes, and kills these capsular bacteria. But post-splenectomy patients lose this protection entirely. That drastically increases their chances of a lethal infection. And this is exactly why post-splenectomy patients absolutely must be immunized against these specific capsular bugs right away. But I've got a surprising, highly testable fact for you to file away. The Klebsiella exception. While Klebsiella is a deadly capsular bacteria totally notorious for causing hospital-acquired infections, there is actually no available vaccine for it. Zero. You can and absolutely must immunize your splenectomy patients against the others, but you literally cannot vaccinate against Klebsiella. As we wrap up this explainer today, I want to leave you with one final thought. If just a single lipid A molecule housed in a microscopic gram-negative cell wall can trigger a cascade that crashes an entire human cardiovascular system, what other catastrophic clinical secrets are these microbes hiding in their structures? Keep that curiosity alive as you tackle your next microbiology topic. Thanks for learning with me today. I'll catch you in the next one.